Good morning, students. We are going to discuss about centrifuge today. So, centrifuge is very much essential in the bioanalytical techniques, in the biological laboratories, and even in the clinical laboratories. Too, centrifuge is very much essential instrument. Now, we are going to see about centrifuge. What is centrifuge, and it is used for what are all the biological functions. So, centrifuge is a mechanical device used for separating substances from the solution according to their density, size, shape, and viscosity of the medium. This is very much important. We can take it down. So, based on the size, shape, viscosity of the medium. So, and another one is density. So, what is centrifugation? Centrifugation is a separation method which uses the action of centrifugal force accelerate in the sedimentation coefficient based on the acceleration forces RCF, that is rotation per minute. And this is how the centrifugation will uh, taken place. Principle of centrifugation. The centrifuge involves a principle of centrifugation where the acceleration at centripetal force causes denser substances to separate out along the radial direction at the bottom of the tube. We can concentrate in the important sentence, that is centripetal force causes denser substances to separate out along the radial direction at the bottom of the tube. So, centripetal force is very much essential to separation. In a solution, particles whose density is higher than that of the solvent sink, and the particles that are lighter than it float to the top. So solvent is higher than the particles, means the particles will sediment, the lighter than it float to top, the greater the difference is in density, it will sedimented in the bottom. So RCF's relative centrifugal force. RCF is the ratio of the centrifugal acceleration at a specified radius and the speed to the standard acceleration of the gravity. So relative centrifugal force is defined as F equal to omega square R. So here F is called the intensity of the centrifugal force. Omega is called angular velocity of rotation. R is distance of migrating particles from the solution. What are all the methods of centrifugation we are available in this technique? There are many types of preparative centrifugation, such as differential density gradient centrifugation and the rate zonal density gradient centrifugation divides into two, that is rate zonal centrifugation and isophyconic centrifugation. So the third one is centrifugal elutriation. So we can see here centrifugation methods. So initially we can take it the sample and the density gradient, you can see here the picture. We can take it the samples and the rate zonal centrifugation, based on the density they can separate out. So in the top we can see sample, and now in between there is a sucrose density layers, and we can see in the right side picture the particles will separate based on the density size wise. Okay. So, in the case of differential centrifugation, the process of separation of cell organelles is known as subcellular fractionation. To isolate a specific organelles, initially the organs, liver, brain, or kidney are homogenized in a suitable homogenizing medium at 4 degrees centigrade. The resulting suspension containing many intact organelles is known as a homogenate. So, we can see this picture. So initially centrifugation occurs at 800 gravity for 10 minutes. The next one is centrifugation at 15,000 gravity per 10 minutes. So the later one is centrifuge supernatant at 10,000 gravity for 60 minutes. And the final one is centrifuge at 200,000 gravity per 3 hours. So here the particles will separate out based on the rotation and the timing. Based on the rotation and the timing, they will separate out. So the initially the suspension of the broken cells containing subcellular components. The next one is nuclear sediment and mitochondria. Cell organelles like mitochondria, lysosome sediments are separated out, and fragments of plasma membrane and endoplasmic reticulum sediment also separated out. And the finally ribosome sediment also separated out. This is how this differential centrifugation taken place. And the next one is a density gradient centrifugation. The density gradient centrifugation depends upon the Bion densities of the particles. This method gives a much better separation than the differential centrifugation. Two types, that is red zonal and isophiconic. So we can see here this picture density gradients. We can take it sperm as a material, and density gradient 40% and density gradient 18%. Post centrifugation, after centrifugation we can take it as seminal plasma separated out, non-motile sperms, dead and other cells separated out, and motile sperms we are seeing under the bottom region. So we can take this motile sperms only for the biological or clinical research also. So the next one is red zonal centrifugation. It is a type of density gradient centrifugation. Differences in shape and density of the particles. So it depends upon the size and shape of the particles. So here sucrose solution sample 55% sucrose and 20% sucrose. So we can take it as red zonal centrifugation techniques, and finally we get the small size DNA molecules at the upper and the medium size DNA molecules in the middle region, and large size DNA molecules in the bottom line. So here based on the size wise and shape wise we can get it in the resultant resultant in the solution. So we can take it as isophiconic, means it depends only upon the buoyant density of the particle, does not depend on the shape or size. So here we only taken as buoyant density. So we can see this picture before centrifugation, sample before centrifugation, after centrifugation, the based on the bion density we separated out the resultant particles. So the next type of centrifugation is centrifugal elutriation. In this technique, the separation and purification of the large variety of cells from different tissues and species can be achieved by a gentle washing action using an elutriation rotor. The technique is based upon the differences in the equilibrium setup in the separation chamber of the rotor. So usually centrifugation we can use too many types of rotors. So swinging bucket and simple rotor, like that we are using the rotors based on what we have actually need the sample types. So fluid fine particles here, and suspension the coarse particles taken out by the elutriation method. So types of centrifuge. The different types of centrifuge we are using in the research laboratories as well as in the clinical laboratories. Centrifuge may be classified into four major groups: the desktop clinical centrifuge, and the high-speed centrifuge, ultra centrifuge, and analytical centrifuge. So desktop or clinical centrifuge is very much useful. We can take it anywhere and we can do it our results. It is a desktop. We can use it in our table. Table itself. High speed centrifuge, the mainly the differences in the higher version, the rotation and the centrifugal force. So high speed centrifuge 25,000 to 30,000 rpm, a centrifugal force of about 90,000 gravity, and refrigeration facility to function in a low temperatures are used for self fractionation. Ultra centrifuge 75,000 rpm with the centrifugal force of 500,000 and gravity. Obviously the rotor chamber is fully refrigerated. So the high speed or high rpm and high gravity we can generate generate so much of heat. So we are using this type of centrifuge in a air cooling room only, that is refrigerated room only we can use ultra centrifuge like that. So analytical centrifuge are more powerful than the preparative ultracentrifuge. So sedimentation of characteristics of molecules based on the size and shapes. So different types of rotors we are using in the centrifuges, that is called swinging bucket rotor and fixed angle rotor, zonal rotor. We can see here the first one is swinging bucket rotor, the right side fixed angle rotor and zonal rotor, vertical tube rotor. So this is the rotor types which we want. Different type of samples we can take it different types of rotors. So applications of centrifuge, we can mainly it is used to only for the separation of the macromolecules and the micromolecules of the biological samples as well as the clinical samples. So separation is a main use application for the centrifugation. This technique is also used in the order to study the analyzed macromolecules and their hydrodynamic properties. Mammalian cells can be purified with the help of special type of centrifuge. Centrifugation is known to have a vital role in the fractionation of many subcellular organelles and membranes. Skimmed milk is a form of a milk that has lower amount of dissolved fats. So regular milk with the help of process of centrifugation. So centrifuge is widely used in the field of forensic chemistry. In the field of technique is employed to detect the DNA samples. So it is useful to identify who is victim in this particular case. So in forensic department also it is used widely. So this is how the centrifuge instrument will use in both biological, clinical, and as well as in the forensic industry we are using so far. Thank you.
Go from Papa to Super Papa.